Welcome to the screencast on blood. If you are not using the revised PowerPoints that I sent out, please press pause and then print those out and use them because it'll make more sense with those. So the first thing you want to know about blood is its basic composition. It is a connective tissue. You may recall that there's a basic definition for a connective tissue. Anybody remember? It is cells suspended in an extracellular matrix. So in the case of blood, we have cells, red blood cells and white blood cells, which we call formed elements, that are suspended or embedded in an extracellular matrix that we call plasma, which happens to be a liquid in this case. So the pH of the plasma is usually somewhere between 7.35 and 7.45, so it's slightly alkaline. You will notice that there are two cell types, and then we have these platelets. They are fragments of cells, which is why we say that the blood is composed of plasma plus formed elements as opposed to plasma plus the cells. And we'll go into later exactly what the platelets are. It'll make more sense. So you can take blood and put it into a centrifuge, a little thing there that spins around real fast, and then separate out the plasma from the formed elements. When you do this, you can measure what's called the hematocrit, which if you look down here, it's just the proportion of your blood or the percentage of your blood volume that is taken up by your red blood cells. There's just a little bit layer of white blood cells there, but anything that's red in this column of blood is red blood cells, and that is your hematocrit. These are one of the few numbers that I do want you to keep in mind. Average hematocrit for males, 47% plus or minus 5. Average hematocrit for females, 42% plus or minus 5. So what then is in this plasmid that we speak of? It is, as the PowerPoint says here, mostly water. Most of your body is mostly water. Plasma is no exception. There are a number of proteins that are floating around dissolved in the plasma that we don't really talk about very much. And then a lot of waste products, lactic acid, other metabolic byproducts. Creatinine, if you might remember, would come from your muscle cells. There's the breakdown product of creatine phosphate. And then we have this stuff called urea, which is one of the major metabolic byproducts of your body. If you recall when we were talking about muscles, you can go aerobic or anaerobic. Well, that's not the right way to say it. When you're thinking about metabolism, there are a number of different types of compounds that your body can break down to convert into ATP. What it prefers is glucose or any sort of hydrocarbon, which is why, or I should say carbohydrate, which is why lipids also work well, because they're carbon, hydrogen, and oxygen. Your proteins have amino acids on them, which contain the NH2 group or the amine group, and that doesn't fit through any of the carbohydrate-friendly ATP-producing machinery. So anytime you break down a protein and use it for energy as opposed to building proteins in your body, it gets deaminated and that amine group gets converted into urea. So it's one of your body's major metabolic waste products. There's also a lot of nutrients that float around in your bloodstream, glucose, amino acids. Then you have things like electrolytes. I've got 3 blanks here. If anybody can think of electrolytes, they might be things like sodium, potassium, calcium, respiratory gases. You breathe in oxygen, you breathe out CO2. And then all of those hormones from chapter 16 are floating around in your bloodstream. So there's all of this stuff that gets transported in the plasma. This then brings us to the formed elements, or all of the cell type things in your plasma. And they are red blood cells, white blood cells, and platelets. All of these pink looking things with no dark purple dots are your red blood cells, aka erythrocytes. And then you have a number of different white blood cells. They all have nuclei That's what makes them different than the red blood cells, which lack the nuclei. And then these here are the platelets, and you can see that they're just little fragments. They're stained dark, but they don't have a nucleus. It's just a whole lot of protein, so they stain dark. The first of the formed elements we are going to talk about are your red blood cells, or erythrocytes. Their job is to carry oxygen, which is what the hemoglobin is for. And in order to enhance their ability to do this, they have two special adaptations. One is that they have dumped their nucleus, and they really have no organelles, so they're essentially just floating bags of hemoglobin. In addition to making the maximum room for hemoglobin inside the cells, their shape, which is called biconcave, because it's concave on top and concave on the bottom, gives them a very large surface area to volume ratio, which makes it very efficient, or makes them very efficient at exchanging oxygen either in the lungs or in your muscles and other tissues that need them. This is what hemoglobin looks like. Each molecule of hemoglobin has 4 peptide chains, that's the globin part, with an iron-containing pigment called heme in the middle. So each of your hemoglobins has 4 of these heme groups. Each heme group can bind up to 1 molecule of oxygen, so each molecule of hemoglobin can bind up to 4 molecules of oxygen. And then just remember, there are countless billions of hemoglobin molecules inside of every red blood cell, but each molecule can carry up to 4 molecules of oxygen. And you're going to see that this is important later when we talk about loading and unloading of oxygen in lungs and tissues. And it turns out that the oxygenation status of these 4 hemes all affects each other, and that affects the ability of hemoglobin to either pick up more oxygen or release lots of oxygen in the tissues. So the next topic we need to talk about is red blood cell production. We will back up a step and first talk about blood cell formation in general. So whether you're talking about a red blood cell, a white blood cell, or a platelet, it's all referred to as hematopoiesis, and it takes place in the red bone marrow. So if you remember, that's wherever the spongy bone is, in the epiphysis and other parts of your skeleton where there's lots of spongy bone, like your hip, the top of your femur. When it comes to forming blood cells, all of your blood cells, all the different types of white blood cells, red blood cells, and the cells that produce platelets, are all derived from the same stem cell, which is called a hemocytoblast. So when people in the news talk about adult stem cells versus fetal stem cells, these hemocytoblasts are one of the adult stem cell populations that people want to isolate and see how many different cell fates they can get it to adopt in order to avoid using fetal stem cells. But in the normal way that your body works, these hemocytoblasts are the cells that, when exposed to different hormones and chemicals, are going to divide and then differentiate into different types of blood cells. If you need to make more erythrocytes, then your body releases erythropoietin. So this is actually released by the kidney. It's a hormone. It travels everywhere in the bloodstream, and when it hits your hemocytoblasts in the bone marrow, it causes the hemocytoblasts to differentiate or develop into erythrocytes. The trigger for it is called hypoxia, which we will talk about in more detail later, but this is a lack of oxygen in a given tissue. So when oxygen isn't making it to the kidney produces erythropoietin, or EPO. When EPO hits the bone marrow, it causes the production of more red blood cells. So your red blood cells do not live forever. I forget how long they live. It might only be like 120 days or something. and they don't have a nucleus, so they can't divide, which means they have to be cleared out of your bloodstream after they've served their usefulness. So this happens in the spleen, is where the red blood cells are cleared from circulation. The iron is salvaged, it's sent over to the liver, and then it's stored there until more heme is made, or more hemoglobin. The heme, or pigment portion of the hemoglobin, is degraded into a bile pigment called bilirubin. That also happens in the liver, which produces bile. So your heme that you're done with from your hemoglobin ends up being excreted with any food that you don't absorb. And then the globin, or the amino acids, are just broken back down and then used to produce other proteins that your body needs. This, by the way, is one of your open-ended essay questions, and how the information you need to know to answer it is all on this slide here. Erythrocyte disorders. So things can go wrong with your erythrocytes, one of which is anemia. Anemia refers to, as it says here, low oxygen carrying capacity in the bloodstream. So it doesn't necessarily refer to the oxygen status, but the ability of the blood to carry oxygen. Let's see, and there are a number of different things that can induce or cause anemia. You could have not enough erythrocytes, and this would be due to hemorrhage, so this would be called hemorrhagic anemia. Hemolytic anemia is when your red blood cells rupture for whatever reason. And then aplastic anemia is when you're not producing enough red blood cells. So what you want to focus on as we go through the slides are the different kinds of anemias, and not necessarily what's after the numbers. So know these three different types of anemia. Another thing that can cause anemia is just not having enough iron. So that is iron deficiency anemia. And then you can have problems with your hemoglobin. One type of hemoglobin problem is called thalassemia, which is just either, as it says here, an absent or an improperly functioning hemoglobin chain, which means you've got a mutation somewhere that's causing either no hemoglobin to be made or a hemoglobin that doesn't function properly. Similar to thalassemia, but for some reason that I don't understand, in its own category is sickle cell anemia. So it is another hemoglobin defect, which causes the hemoglobin to be unable to bind oxygen very well, and also causes the cells to take on this sickle shape. So it's interesting, at least to me, you have this one substitution or mutation where normally you have glutamic acid, and that gets substituted with valine, and you go from normal hemoglobin and a normal shaped red blood cell to abnormal hemoglobin, which somehow affects the shape of the entire red blood cell. The last type of anemia is called pernicious anemia, which is caused by a deficiency in vitamin B. So that is the first half of the blood. Stay tuned for one more screencast over the second half of the blood.